We are going to do the final topic of the three topics which comes together, that is the cell cycle, cell death and cancer. We did cell cycle, we completed cell death, and now we are moving to cancer. But prior to getting into cancer, one thing should be kept in mind, that you should be thorough with your signaling cascades operative within the cell that we learned in our syllabus, as well as we should also keep it in mind the tumor suppressor genes that we studied in cell cycle. With that, we start with cancer. Now, the origin of the word cancer has now become a very common term because in recent times, plenty of people suffer from one form of cancer or the other. Now, this word was originated from the classical, had its origin, just a second, from Hippocrates and Galen. They were the classical Greek and Roman physician, and it was them who laid the foundation of modern medicine. The name cancer was given by Hippocrates. The ancient origin of this word comes from the word carcinos or carcinoma, which is ancient for crab. Now, cancer is the Latin equivalent for this carcinoma. And further, in the earlier times, Galen performed some early surgical interventions of cancer. And he had postulated that this disease is something which is best left untreated. In fact, in a lot of cases in present-day cancer, when the malignancy, when the cells have become malignant, and come to the concept of what is benign and what is malignant. But in present-day concept, when the cancer has become malignant or the cancer is in the late stage, even the physicians also advise it's preferable not to do anything. Just go home, and it's almost waiting for death, but still wait for death. That is the best option. And it was Galen who also believed that the disease resulted from the imbalance in the four bodily humors. Now it was the blood, phlegm, yellow bite, and black bite. So this was I just wanted to show you that how far the origin of cancer dates back. When you look into the different important dates in the discovery of this field, there are two particular dates I'd like to highlight. I'm not getting into the details of all the dates. One was the discovery of the Rouse sarcoma virus by Peyton Rouse in 1911. In 1911, for the first time, Rouse showed that there are viruses which are capable of oncogenic transformation of a normal cell to a cancerous cell. So that was one important discovery in the field of cancer. And then there came another important discovery in the field of cancer in 1971, when it was seen that the growth of tumors is favored by angiogenesis. Now, angiogenesis is a molecular phenomena of blood vessel formation. So it was Folkman whose experiments provided the evidence that whenever there is an abnormal cellular growth, which is nothing but cancer, then blood vessel formation is required by those cells. And you can assume why blood vessel formation is required. Because whenever there is a group of abnormal cells developing in a particular part of our body, you know that all cells require oxygen for survival. So if a mass of cells is, if a group of cells are suddenly dividing and proliferating, they need oxygen. And if this mass of cells is somewhat distant from any blood vessel, then this mass of cells will secrete molecules which will attract the sprouting of new vessels from the old vessels. And these new vessels will sprout out from the old vessels and will move towards this mass of cells, so that through these new vessels, blood comes, and from the blood, these mass of cells can take in oxygen. So this was an important discovery in 1971, where it was evidenced that angiogenesis fuels tumor growth. That means it is a mutual process. The abnormal cellular growth initiates the formation of blood vessel towards it, And once blood vessel is formed towards it, as you can see, blood can now move to the areas where this mass of cell has developed. And these cells can then take in the oxygen coming through the blood. Later on, there are a lot of other experiments, each equally important. But the first, another important discovery in 1979 was done by Pierce, where he said that the microenvironment of the cancer cell is very important. So these were some of the seminal discoveries in the field of cancer. In 2005, Lee and Henrik Mellon used the zebrafish model, and Leiden in 2005 discovered the pre-metastatic niche. Niche means an area or place or the environment, local environment. So these were some of the important discoveries in the field of cancer. And now I move on directly to the tumor microenvironment. Now, when I'm using the word tumor and when I'm using the word cancer, these two are not always synonymous. A tumor, as you can understand, is a group of cells that has started dividing uncontrollably. It starts with a single cell. A single cell is mutated. You have studied mutation. And as a result of this mutation, the single cell becomes aberrant. It becomes defective. Somewhat, some way, it is able to bypass the restriction checkpoints present in our cell cycle. So it can evade the restriction checkpoints. It is also capable of evading the apoptosis phenomena. These are two normal phenomena to keep the cell in check. So whenever there's a cellular aberration, either there's a cell cycle checkpoint that arrests the cell, instructs the cell to die by apoptosis, or repairs the cell. So if a cell is aberrant and it survives, then we have to assume that it has escaped the cellular checkpoints. It has also escaped apoptotic death. So the moment a cell is able to escape the cellular checkpoints as well as apoptotic death, it is imperative that the cell becomes to some extent immortal. The cell can now not be easily killed by the molecular cascades that are programmed in it to kill it. And now this cell, as it has evaded apoptosis, it has evaded the cellular checkpoints, it comes out of the strict control of cell cycle. The cell becomes independent of growth factors. If you remember your class in cell cycle, you must have understood the importance of growth factors for passing of a cell from G1 to S. Now, whenever a cell becomes immortal, a cell escapes the cellular checkpoints as well as apoptosis, it also escapes its dependency on growth factors. So the cell is not dependent on growth factors anymore. So if there is present or absence of growth factors, it doesn't matter. The cell moves out from M, passes through G1, S, G2 without any hindrance. And this cell then, as I said, starts dividing, proliferating rapidly to give rise to a mass of cells which are also very much defective like itself. So now we have a mass of cells dividing in our body. These mass of cells are defective mass of cells. As long as this mass of cells are localized into one part of the body, they have not spread, they have not moved anywhere. These mass of cells are referred to as benign and they are referred to as tumors. So it's a benign tumor. That is, they are restricted to only one part of the body. They have become aberrant. They're resulting in abnormal growth of mass of cells, but they have been restricted to a part of the body. Now, as proliferation of this mass of cells proceeds, some of the cells gets further mutated, gets some additional property, develops some additional property by which they can come out from this mass, move into the blood vessel, travel through the blood, escape from the blood vessel into a fresh area where they again start dividing. So let us suppose the initial infection occurred in the kidney. So there is a mass of cells localized in the kidney. Now, as I said, as the replicative multiplication of these cells proceeds, some cells gets further mutated. And one of the cells from this mass of cells then enters blood vessels, travels through blood, and escapes the blood vessels in the liver. And in the liver, this single cell again starts dividing. Now, whenever a single cell moves from a kidney tissue to a liver tissue, you should understand that they develop certain properties of abnormality and they lose certain properties of being normal. And that is why they can move from kidney to liver. And now this mass of cells have infected the liver, that single cell infected the liver. And within the liver, this mass of cell is again dividing. So the moment a mass of cell dividing in a body part such as kidney develops such a capacity that some of the cells can escape the kidney through the blood and now go to any other organ of the body and then enter that organ and start dividing again. This phenomena is known as metastasis. Those tumors which are capable of metastasizing is known as malignant tumors. And the cancer name is given to only those tumors which are malignant in nature. So that was basically the understanding of the terminology of why we sometimes refer to it as tumor or sometimes refer to as cancer. But the word cancer means the mass of cells have become malignant. So it's a word basically which gives us a psychological depression. So whenever you hear this word cancer, but whenever you hear this word, this line, specifically those who know about this disease, they hear this line that she is suffering from cancer, is basically, it gives a very psychological impact. Because this is a disease which has so many facets, it becomes very difficult to control, as this disease involves a lot of molecular cascade. And let's look into the basic properties of this basic disease. Now, since I started describing tumor and cancer, by now you have understood that unlimited multiplication. These cells have become mutated. They have escaped the mechanism of cellular checkpoints. They have escaped the mechanism of stringent control over them by the cellular checkpoints. They are independent of growth factors and hence they have escaped growth suppressors. They can promote invasion and metastasis. So a mass of cells dividing in one part of the body at a time, if develop certain properties, can escape that area through the blood vessel, enter blood vessel, and through blood can go and infect a new area. So that is invasion and metastasis. I started saying that they can resist apoptosis. So this mass of cells somehow do not allow either the intrinsic or the extrinsic pathway of apoptosis to operate within themselves. They can stimulate angiogenesis. Angiogenesis is the formation of blood vessels. So for their need of energy, food, and oxygen, this mass of cells can stimulate angiogenesis. They can constantly induce proliferative signals within themselves so that the cells constantly go on dividing and dividing. See, elimination of cell energy limitation. Every cell requires energy, but these cells, and once that energy is drained from a cell, the cell stops dividing or the cell is exhausted or the cell is allowed to die. But they have overcome this property also. They do not require as much energy as that of a normal cell. And whenever there is an energy limitation, they do not become quiescent. They can continue dividing. So they have overcome this problem also. They can evade immune destruction. Remember, whenever a cell is mutated and aberrant, that is now no more a normal cell of a body. So immune cells of a body recognize these cells and kill them. That is the normal function of immune system. We have a very stringent immune system occurring in our body. These cells are also capable of evading the immune system and evading the immune system mediated destruction. And obviously, since there is mutation and genetic instability, these genome instability and mutation in fact gives rise to abnormal cell from a normal cell. So these are the different widely recognized properties of a cancer. And this is just to give you a glimpse about the human genome and mutation rate. The human genome has 3.2 million base pairs of DNA, and the mutation rate is estimated to be 1 nucleotide change per 10 to the power 9 nucleotides per DNA replication. So keep it in mind that a silent mutation rate is constantly occurring in our cell during every cycle of replication. As long as this mutation doesn't change a sequence that codes a different amino acid, that there is not much of a problem. But the moment this mutation results in a change in the coding sequence, which results in a change in a different, in the production of a different amino acid, then only there comes a problem. Now the genome, as I said, the genome is altered on an average by 3 nucleotides each cell cycle. And this alteration can take place in the non-coding region of a DNA. It can take place in the coding region of DNA. As long as it is taking place in the non-coding region of the DNA, it's not much of a problem. But if it's taking place in the coding region of a DNA, keep it in mind that when I'm talking about the coding region of a DNA, the coding region of DNA includes the promoter elements, the upstream enhancer or the repressor elements, the downstream coding sequences, and the terminator. So if there is mutations in these regions, then obviously these mutations might have drastic effect leading to the transformation of a normal cell to an abnormal cell. Now, there are 30,000 genes in humans. You can understand the volume of genes present in our body, and alternative splicing can give rise to about 120,000 cDNAs. So you can understand that 30,000 genes gives rise to about 120,000 cDNAs. That means there are so many amount of mRNAs being produced in our body. So you people can be aware that at the molecular level, the volume of work the cell is doing and the volume of mRNA that is being generated, and accordingly from this transcripts of 1,20,000, there are about 2,50,000 proteins in humans, and these proteins can further undergo post-translational modification. If you remember the molecular pathway of central dogma, there was this replication, transcription, and translation. In translation, these proteins are produced, and these proteins then undergo post-translational modification, and you can understand that it's around 10 lakh modified proteins available in the body. So when we have so much amount of voluminous protein products from 30,000 genes, there is a place where mistakes or errors may get into. If the error is taking place in a coding region of a DNA, the error becomes a vital error. But if the error takes place in the non-coding region of the DNA, then these errors do not so much affect the life of a cell. So coming to once more that how this cancer, now the oncogenesis, carcinogenesis, these are different ways to describe a malignant state. Tumor, benign or malignant. Tumor, isolated group of cells growing in a body. That's benign, that is a tumor. Whenever cells migrate from the original site to different places, that becomes a malignant. Okay. Now, the subject of studying cancer can be referred to as oncology. The subject is referred to as oncology. The process in which a normal cell gradually becomes cancer cell and then a cancer cell starts proliferating to give rise to a growth of cancer is known as tumorigenesis or carcinogenesis. Genesis means development of something. So it's carcinogenesis or tumorigenesis, okay? So as I was saying that oncogenesis, that means the development of cancer may be due to spontaneous genetic changes such as gene or chromosome mutations. They may be due to spontaneous means that is going automatically within your body, because just now I said you that there are silent mutations occurring in our body. It may be due to exposure of mutagens or radiations. So if we are exposed to certain radiations, to certain chemicals, then these chemicals might induce mutations in our cells, in our DNA. And these mutations might result in lesions in the DNA. DNA might be damaged and a cell may become cancerous. And as I told you in one of the earlier slides, that there are certain viruses, tumor viruses, which are capable of resulting in human cancers. One such is the papilloma virus that results in the cervical cancer. Now, at the initial stage, while I was explaining you the terminology for tumor and cancer, I said that if the abnormal cells remain at the originating site, it is known as a benign tumor. Benign tumor does not cause serious problems because they're restricted to a local site. But once the cell leaves the original site as a result of further mutation and spread to more organs and spread throughout the body, in this process, it has developed the property of metastasis and the cancer has become malignant. So we discussed this. Now, this is a mammogram showing a tumor. You can see the growth of a tumor. Now, normal versus abnormal. If you look into the two pictures, this is a normal skin and this is a tumor growth. You see that this is the underlying tissue, a basal lamina. Now, this is the basal layer of dividing cells. Cell division in the basal layer is giving rise to new cells, as you can see, that migrates outward. The more these cells are migrating to the outer surface, they are losing their property of division, but they are getting differentiated. Keep it in mind. So this is the layer of cell, that is basal layer, that is containing the dividing cells and the non-differentiated cells. But as the cell division proceeds and the new cells are rising, they migrate to the topper region. And as they migrate, they gradually lose the capacity to divide and they become differentiated. And now look into the right side picture. In tumor growth, this orderly process is lost. As you can see, some cells have become mutated. And these cells, once they have become mutated, they start dividing. One mutation leads to another mutation. You can see there are two different colors. So there is a second set of mutation and some of the cells then starts migrating to the outer areas. But keep it in mind that these cells which starts migrating to the outer areas, they still retain their dividing capacity. Here in the normal case, what we see that when the cells are migrating to the outer areas, they have lost their capacity to divide, proliferate. But in case of a cancer, whenever these cells have become abnormal and further mutations results in further abnormality, these cells gradually also can able to migrate to the outer layer, but these cells have retained their capacity to divide, but they are not differentiated so much as compared to a normal cell. So as a result, what you can see is that there is an excess accumulation of dividing cell occurring place. And this results in somewhat a tumor growth. So this is just a simple comparison. Now, if we talk about the benign and the malignant tumors, based on which area, which tissue of the body has giving rise to that tumor mass. They can be of three categories. Carcinoma, they arise from the endoderm or the ectoderm. That is any cancer related to skin, neural epithelium, or the intestine. They are known as carcinoma. Sarcomas arise from mesodermal tissue, that is blood related, connective tissue and muscle. And then you have leukemias and lymphomas. These are cancers of the blood forming cells. That is, these are cancers of the cells which forms the immune system of our body. When we talk about the basic step, just a second. In tumor formation, there are 4 basic steps in tumor formation. The first is that they have shown you the epithelial cells here, as you can see. The first step, there's a blood vessel being shown. So the first step for a normal cell to become a cancer cell is a mutation. The mutation may be in intrinsic nature, that is, it has developed from within due to a certain abnormality. It may be as a result of exposure to a physical or a chemical mutagen. Once the cell is mutated, you can see that the mass of cells have started dividing. The tumor cells have started dividing. They lose their differentiation property. They gain their hyperproliferative property. And what they do, they secrete factors that start attracting blood vessels towards them. So they prefer or they initiate the process of angiogenesis. The moment the blood vessel sprouts and moves towards them, some of these cancer cells might enter the blood vessels and through blood may travel to any distant tissue of the body. That means intravasation, entering the blood vessels and traveling through the blood vessels into a tissue space where extravasation occurs. That means it comes out of the blood vessel. And then in this fresh site, you can see the cells have moved from this to, the cells have moved from this to this site. In the fresh site, the cells come out of the blood vessels and these cells again then starts dividing rapidly. So these are the 4 basic steps in development of a tumor, rather in development of a metastatic tumor. Because if a tumor is localized, then 3, 4 doesn't occur, 2 also doesn't occur. Angiogenesis is preferred, but not to such extent. But for a cancer which is malignant, as you can understand that all the 4 steps of cancer formation occurs. Now coming to the multi-step nature of cancer. Now, this is a model which is known as the Walgenstein model of the formation of colorectal cancer. Now, in this model, you will be able to understand that one mutation is not always enough for a normal cell to become an aberrant cell. Multiple mutations are required for a normal cell to become an aberrant cell. This is a case of colorectal cancer. So you can see the site of cancer is here. Initially, what happens? There is a mutation occurring in the APC gene in chromosome 5. The full name of this gene is adenomatous polyposis coli. Okay. Now, once there is a mutation in this gene, both the alleles, there is increased cell growth. But this cell growth is benign in nature as the cell growth is restricted to the colon area. So there is a small benign polyp with the loss of adenomatous polyposis coli gene. Next, after this, there's a second mutation that occurs within these cells, transformed cells. This mutation, you have heard about this protein, you have heard about RAS when we did the cell cycle and when we did the growth factors. If you remember, RAS is an important molecule that plays a huge role in a lot of cellular signaling processes that finally culminate in the production of cyclin D. Now, chromosome, whenever there's a mutation in chromosome 12, the RAS proto-oncogene, I'll discuss why it's called a proto-oncogene later on. The RAS proto-oncogene is mutated, and as a result of the mutation of a RAS proto-oncogene, we get a second class of tumor, that is class 2 adenoma. After this, there are two vital mutations occurring. One mutation is the loss of both copies of a gene named DCC on chromosome 18. Q, 18q means P and Q, the long arm of chromosome, 18q. And followed by the loss of chromosome 18q, we see that there is a larger adenoma, that is class 3 adenoma. So you can see one mutation was not enough to transform the normal cells into a colorectal cancer. It started with the loss of a single gene APC, followed by the mutation in the RAS proto-oncogene, followed by again a mutation on the DCC, which is a tumor suppressor gene. Till this much time, as the story went, the tumor is restricted to the colon. The tumor is still benign. It has not spread out, it has increased in size. But there is the final mutation with the loss of P53. And understand that once P53 is mutated and we lose the normal function of P53, you have studied P53 in apoptosis, you have studied P53 in cell cycle. So the cellular control on these cells is totally lost, and this tumor mass gradually develops property by which it becomes a malignant tumor mass. These malignant cells are now able to travel via blood vessels to any different part and result in the growth of a abnormal tissue mass in another tissue site of the body. So this is a model form of colorectal cancer. This model I just described to explain you the fact that one mutation is not enough to transform a normal cell to an aberrant cell. Multiple mutations in the process are required, which converts a normal cell into an aberrant cell. And of these multiple mutations in 99.99% of cancers, it has been seen that two very vital genes are constantly mutated. One is the P53 and the other is the tumor suppressor gene, retinoblastoma. So in most cancers still that have been studied, the retinoblastoma gene or the P53 gene is always mutated. So we have a question, I guess, from Kushal. This is a very simplified diagrammatic steps of metastasis. You can see normal epithelium, as you can see. Now you can also see that a benign tumor formation is occurring. That is, a cell has already got mutated, and as a result of mutation, a cell has become abnormal, and abnormal growth of cell has occurred. So benign tumor formation has occurred, and these tumor cells now secrete substances which results in sprouting of blood vessels towards it. So you can see blood vessels have moved towards this benign tumor mass, and some of the cells can enter the blood vessels, a phenomenon known as intravasation. And once these cells have entered blood vessels, they are traveling through the blood vessels, as you can see. And then in this case, they have shown that they travel to the liver. These cells are adhering to the inner surface of the blood vessels once they have reached the liver. And gradually there is extravasation. The cells comes out from the blood vessels and invade the tissue area of the liver, and within this liver tissue, the cells now colonize the liver and gradually starts dividing to give rise to full-grown tumors. So this is a very simplified concept of metastasis, but remember that whenever there is a metastasis occurring, this occurs in a stepwise manner. the cancer cells at the first instant attacks the basement membrane and reaches the blood vessels, as you can see, by using the extracellular matrix. Now, the cancer cells can be attracted to by signals such as the epidermal growth factors. So you can see that some growth factors are being liberated, as a result of which the cancer cells attack the basement membrane and are reaching the blood vessels. Here they penetrate the blood vessels, and as you can see, it has been shown that how the cancer cells are entering the blood vessels. Let's just look at the picture. We're not going to get into the details of this picture today, or in your, or particularly in the undergraduate classes. So this is a summary of this simplified way in which a metastasis is occurring. Now coming to the causes of cancer. As I said, cancer can be caused due to spontaneous mutations. They can be caused due to viruses, or they can be caused due to certain physical or chemical agents. So those agents which are capable of causing any sort of mutation are known as mutagens. Some mutations might lead to cancer. Remember, all mutations do not lead to cancer. Some mutations might lead to cancer. So those mutations that lead to cancer, those chemicals or mutagens are known as carcinogens. So there's this beautiful line that says that all carcinogens are mutagens, but all mutagens are not carcinogens. Explain. I'm repeating once more. All carcinogens are mutagens, but all mutagens are not carcinogens. That is the line. As I said, anything that can cause cancer is known as anything that can cause mutation is known as a mutagen. Those mutagens that can cause cancer are known as carcinogens, but all mutagens do not cause cancer. So they can cause a different category of mutations, but they do not cause cancer. So only those mutagens that can cause cancer are known as carcinogens. And as you can see, they can be divided into physical and chemical carcinogens. Now, in case of chemical carcinogens, alkylating agents such as azo-dyes, polycyclic aromatic hydrocarbon found in smoke from wood, coal, and cigarettes. So when you inhale these polycyclic aromatic hydrocarbons, specifically from cigarettes, those who are smokers often have this phenomenon of lung cancer. But it's a melancholy that all active smokers do not suffer from lung cancer. There are a lot of passive smokers also. If there is in anybody's house, a family member is smoking profusely, and if the smoke is inhaled by the other members of the family, so any other member can also develop lung cancer. Now, as I was saying, that this polycyclic aromatic hydrocarbons found in smoke from wood, coal, and cigarettes can lead to mutations in the cell, lung cell, resulting in cancer. Then there is this natural metabolites like aflatoxin from fungi, if taken in might lead to cancer, nitrosamine. So these are carcinogens. Tobacco smoke is an active form of carcinogen because it contains polycyclic hydrocarbons. Now coming to the physical carcinogens, the source of physical radiations are sun, cellular telephones, radon gas, electric power lines. But it has been seen that ionizing radiations are more in fact capable of causing cancer, because ionizing radiations can lead to the production of ions in your body. So it is this ionizing radiations that do cause cancer. For example, mutations from radiations in UV light, UVA and UVB, specifically UVA, plays a role in skin cancer. So these are some of the physical agents that cause cancer, that is the radiations, the UV rays. Nuclear radiations also can lead to skin cancer. Nuclear radiations can in fact lead to any form of cancer, because whenever there is a nuclear catastrophe, if you remember the story of Hiroshima, if you know the story of Chernobyl, or in 2 to 3 years back, there was this again a nuclear catastrophe occurring in Japan. So nuclear catastrophe, whenever this nuclear, I mean to say, isotopes enter our body, they results in production of whenever this gamma rays or alpha rays or beta rays enter our body, they result in production of damaged DNA. So as a result of which, any form of nuclear catastrophe do results in huge amount of cancerous conditions in the population. I don't know if you all know it or not. People who suffered the nuclear bombing at Hiroshima and Nagasaki, generations after generations, those who survived, those who died, nothing can be told about them, but those who have survived, their generations after generations have some form of abnormality in their body. And now coming to another category of molecule that can cause cancer are tumor promoters. They are not the direct causative agent of cancer, but they help a proliferating group of cells to become a malignant group of cells. And one such example of tumor promoter is estrogen itself. So we'll stop our class here today.